My name is Gustavo Mendes, and I'm here as a fellow in regulatory affairs at Simulation Plus with Xavier, Pepin, that will also present as part of this real-world impact and regulatory decision-making track. And Xavier is going to have a presentation entitled "The Advantages of PBBM and PKPD Modeling to Define the Solution Safe Space." Another presentation that is related to this track that we are discussing on case examples and applications of this modeling and simulation. So Xavier is Associate Vice President of Regulatory Affairs at Simulation Plus. So thank you very much, Xavier, for accepting to present here, and I give the floor to you.
Thank you, Gustavo. Thanks for the invitation and you're moderating this session. So I'll share my screen. I hope you guys can see it.
I'll let you know when it's it.
Can you see my screen? Yep.
Not yet. Now, yes, yeah.
Right.
But just we are not seeing the PPT. We are seeing only your screen on the platform.
My presentation.
No, not the slides.
Okay, sorry about that. Stop sharing.
I think you have to share the full screen, I think.
Chrome tab, entire screen, okay. Okay. Do you see my screen now?
Yes. Now we can see. Yeah. Thank you.
Okay, fantastic. All right. So thanks for the kind introduction, Gustavo. I'll start with the first slide to illustrate the advantages of physiologically based biopharmaceutics models in the industry. First and foremost, it brings the scientist with a mechanistic understanding of what is limiting absorption, whether it is dissolution, permeability, precipitation, and that mechanistic understanding is then applied to batch performance to help justify key specifications for critical quality attributes. And of course, this can be used as part of the dossier to justify at time of submission, but also post-approval. You can modify those specifications if your batch performance, if your process performance is shifted during batch manufacture. And the use of PBBM is also very important when you request for a regulatory flexibility, because you can define a safe space and move in that safe space your justification, your specification, and they will be justified. And eventually, it's going to reduce the number of unnecessary human testing, so you can apply PBBM to waive clinical trials, and this is used more and more to bridge formulations. So it's very important. So why do we want to use PBBM and PKPD? So first of all, PBBM links, as I just said, the drug product quality attributes to in vivo exposure, and this is applied to many different attributes. Could go from in vitro dissolution to polymorphic impurities to manufacturing process conditions to in vivo degradation, and even in some cases to the effect and impact of the level and nature of excipients, which could play on solubility, on GI transit, etc. So PBBM is a key tool for formulation quality, and that's the first step that will bring those quality attributes into predicted exposure. So we can define a safe space with PBBM, and this is typically done through virtual bioequivalence, where you do try to find the age of failure. So when do you start to fail bioequivalence between a reference batch and a test batch? And for this, we can use either standard bioequivalence criteria or reference scale B criteria if you have a highly variable drug. But the limitation of your safe space will be just based on bioequivalence. If you marry PBBM and PKPD, you can do much more. So PKPD will link your exposure to efficacy and toxicity, and these models are typically based on observed data. So you link your efficacy data, your tox data to exposure. And this can be done through clinical endpoints that are measured in the clinic, or the biomarkers of efficacy. If you have a target engagement that you can measure directly, then you can use that biomarker to justify the efficacy as well. They can be simple mathematical relationships, or they could be mechanistic model based on the mechanism of action and the cascade of biochemical reaction that are happening after the target engagement. So PKPD would allow you to also define a safe space based on efficacy and toxicity, where you would expect that all the batches are we're going to give you the same biological response. So this safe space may be larger than the bioequivalent safe space you get through PBBM. And so it's really important to, when you can to combine these two modeling approaches, because the safe space can be enlarged. So just to illustrate this point, I will take two case studies, industrial case studies. The case study one is for a new tablet formulation of acalabrutinib. It's a salt tablet, so from the salt of that free base. And I will show you how the PKPD and PBBM were used to determine the product dissolution safe space in the target patient population. And the second case study will be another salt for drug X. This model was developed, the PKPD model was developed for this drug, which was poorly absorbable from the GI tract. And in combination with PBBM, we used that model to evaluate the risk for a delayed tablet disintegration. So the first case study is the acalabrutinib maleate tablet. So acalabrutinib itself is a free base which has a very strong pH solubility profile. It's very dependent on the pH, and when the drug is dosed with acid-reducing agents, there is an issue with exposure. The exposure goes down. And since the target patient population in heme cancer are estimated to take acid-reducing agents due to their side effects or co-administered administered treatments, it was really important to manufacture a new formulation which could remove that label restriction for that drug. Maleate salt increases the surface solubility of the drug compared to the free base, which leads to a more complete and more rapid dissolution in all media tested. And you can see here the tablet in green and the capsule, the free base capsule in blue. You have a very clear benefit of the tablet, the salt formulation in terms of dissolution rate. So the model purpose was really to help justify a dissolution specification for that new tablet. So the strategy that was applied for modeling, it was to reuse a previously published PBBM for the free base, which was published here. So you've got the reference if you're interested, and adapt it to the maleate tablet by changing, of course, the pH solubility profile of the drug, because it's a new drug substance, and also using inputs that are adapted to the maleate tablet. So we use the PPSD approach to fit the in vitro dissolution of the tablet and integrate it into the model. The model was validated based on the result from one clinical trial where multiple formulations were tested, including an oral solution. And then the model was also used to predict exposure of virtual batches of maleate tablet to try to find the failure for the safe space. So we used PBBM to run this analysis, but we also used a PKPD model to show that the exposure, or position the exposure of those tablets in a space where the drug was effective based on BTK occupancy, which is the direct blood marker for the drug engagement. So in terms of adaptations, as I mentioned, we integrated dissolution using PPSD approach, and you have here the 2 steps of this method. The first one is to extract the product particle size, which represents the effective API available for dissolution. And here we had 3 different batches with drug substance of increasing size. So you have different batch particle size distributions. And then the second step is to verify that the model can predict dissolution in other conditions. So for these 3 batches, we predicted from purely bottom up the dissolution in different media. And you can see here the fold errors, average and absolute average fold errors are very good. So we've got an accuracy of 6%, so only 6% prediction error in the for that prediction of the pH. So to compare the PBBM validation for the maleate compared to the freebase, we also plotted the predicted versus measured Cmax and AUC. So in blue, you have the previous model for the freebase, and in orange, you have the maleate. So you can see it very well aligns with the previous prediction, but it's adapted, of course, to the tablet performance. So this is for the model validation. In terms of model use, as I mentioned, we use the PBBM to predict the age of failure for the batches. So we had the clinical batches here in green and blue, and we tested 2 virtual batches, virtual batch A and B, which are in purple and red, to just artificially slow down dissolution and try to find where the age of failure was situated. And so these virtual batches A and B were integrated in the model, and we predicted exposure. So you have here the Cmax ratio on the right, and the AUC ratio when the stomach is acidic. And you can see very well that we are anticipated the same exposure for both these virtual batches compared to the clinical reference in acidic conditions. So very, very limited impact of dissolution in acidic condition. Of course, the solubility is very high for that drug in acidic condition. But because this drug is intended to be given to patients that are taking PPI, we also simulated the exposure at stomach pH 6. And you can see here the Cmax ratio and the AUC ratio are clearly showing a slope. So they are going down as the dissolution goes down. And this is represented as a function of percent dissolved at 20 minutes. This is a very practical way to show the data because we want to justify for dissolution. And we use the 20-minute time point for dissolution to show that how this Cmax ratio is evolving as a function of dissolution. So in terms of Cmax, you can see that virtual batch A would not be bioequivalent to the reference in acidic condition. So what I forgot to mention is the reference was kept in acidic condition to calculate the Cmax ratio. So we are shifting everything down because of that reference TAB in acidic condition. Even in terms of AUC, only the virtual batch A would be within the bioequivalence space, but because of variability in the virtual B or in the clinic, that virtual batch A would still not be bioequivalent to the reference because of the variability. So in neutral stomach pH, the safe space that would be offered by just bioequivalence is too small. It wouldn't help and justify specific. So what we did to follow up is to, instead of taking the Cmax ratio or the AUC ratio, we looked at the actual AUC and actual Cmax that were given by those batches in neutral condition in pH 6 in the stomach. And we compared these to the PKPD model that was developed over 2 clinical trials in healthy volunteers and patients. And you can see that these the open symbols, which are blue, the blue symbols show the maleate tablet batches, and the black open symbols show the exposure. The BTK occupancy at steady state versus Cmax or AUC for observed in the clinic. And you can see that these the virtual batch A and virtual batch B would lead to very effective target engagement, because these target engagements were seen in the clinic in the pivotal efficacy study. So we know that they are effective, which means that even though the Cmax ratio and AUC ratio would lead to conclude that we're not bioequivalent to the reference batch, the actual absolute exposures in a pH6 would still lead to efficacy. And so that argument was used to say, to justify for the safe space, we use virtual batches, we use the PBBM to predict exposure and relative and absolute exposure, and then we use the PKPD model to define the safe space. And basically that argument was used to justify that a Q of AT between 20 and 30 minutes would be anticipated to be safe and effective for the 100 milligram acalbutrin maleate tablet. This was leading to the same target engagement. We also tested in the clinic an oral solution, and that justifies for the upper edge of the safe space here. It was tested in the clinic and it was found bioequivalent to the solid formulation. So in terms of dissolution, even the solution 100% dissolved at time 0 would lead to safety and efficacy. So that's all for the maleate tablet. I'll move to the second case study, which was as I said, a poorly absorbable drug. So this drug is administered as a salt, and the salt has a lower solubility than the free base up to the pH max, and then a higher solubility. So it does supersaturate at one point when moving from the stomach to the intestine, and it does precipitate in vivo and in vitro. As I said, the drug is minimally absorbed, so the drug X cannot be followed in the plasma. All the concentrations are below. However, there is a small amount that gets in and is quickly converted to the inactive metabolite, which are called M. In the gut, and especially in the feces, you can follow concentration of a biomarker, which is a direct result of the pharmacological action of that drug. And the idea of the model was to use all the clinical data for the PK, systemic PK metabolite concentration, and also all the clinical data for the concentration of the biomarker B in feces, to build both a PBBM and a PKPD model to support the question that was asked. And this question was around the variability in tablet disintegration, which was delaying a little bit the in vitro dissolution, and we wanted to evaluate that impact in terms of efficacy. So the modeling strategy, I'll go very quickly, not into the details, but we had more than 10 clinical scenarios for the PBBM and more than 10 clinical scenarios for the PKPD model. So it was quite rich data. There were 3 types of formulations. There were salt in a formulation. There was a tablet with the salt and the tablet with a free base that were tested in the clinic. The issue with the PK of M, of the metabolite, it was mostly sparse PK. There were very little studies with full PK profiles, but there was clinical data for the drug efficacy, so the stool biomarker concentration of the biomarker B. And of course, in parallel to all these modeling activities, there was a root cause analysis to look at what could cause that delay in tablet dissolution, which was linked to the disintegrant performance. So in terms of input and model parameters, we had the same approach. We used a mechanistic model for the dissolution. So we used the PPSD model by looking at the solubility and in vitro and extracting a batch representative input for dissolution. We also integrated mechanistic nucleation and growth model, which is a very interesting feature in GastroPlus, because you have one set of parameters you need to define, and then you can keep it constant for all your simulations. And here, for example, you can see the difference between fasted and fed state. So in red, you have the mass of drug that dissolve over time. So you can very clearly see in the fasted state, you reach a maximum and then it precipitates, and that limits the extent of absorption for that drug. But in the fed state, the dissolution is taking more time. The supersaturation is less important, and the precipitation is less important. So you can absorb more drug than in the fasted state. So that is quite interesting, and to be able to predict this. In blue, you have the metabolite plasma concentration, which was fitted in the PBBM part of this exercise, and predicted as well. So in terms of PKPD model, we looked at the luminal concentration of drug X, which, as I said again, is minimally absorbed, and we tried to correlate this with the biomarker B. The biomarker B concentration. So we hope to define a direct relationship, and this was the case. And because of the mechanism of action of that drug, the best correlation was found by looking at the average drug X sequence concentrations and the stool concentration in the biomarker B. A straight line, which was very surprising, and it was not a straight line for any other compartments in the GI tract. So that's really the site of action is probably related there. Interestingly, when we explore the PKPD model, we had different ethnicities, we had different studies, different doses, and it looks random here, but there are actually very nice correlations to be shown. So I'll go quickly through these. First, the dose, it was nicely correlated. So we had a clear dose relationship. Some placebos were also administered and had increasing dose, and the PD model, I mean, the stool concentration of biomarker B was very well correlated to the concentration of the active of the parent. Of ethnicity, we also had the chance to have the same formulation tested in different ethnic groups, and we could see very clear differences. And initially, that was something that was very hard to understand without any PBBM. But when you look at these ethnicities and when you compare them, the ethnicity X had a smaller cecum compared to Caucasian, and that led to an increased drug concentration and therefore an increased efficacy for these ethnic groups at the same dose. The formulation effect was also very interesting to look into, because there were, as I mentioned, 3 types of formulations tested, and some were supersaturating and precipitating, others were not because they were solubility limited. And so in terms of impact on the dose dissolve, that had a dramatic impact. Basically, the salt tablet was the one that dissolved the most, and even though it precipitated, the amount eventually dissolved and absorbed was quite high. The salt in additional formulation had half of the dose dissolved compared to the salt tablet, because it was solubility limited and dissolution rate limited. Sorry. So it was limited. The absorption was much slower, and the dissolution was much slower. And the 3-day tablet was a different form, so the solubility was much lower. Absorption was as well very prolonged compared to the. And these ranked very nicely with the efficacy that was measured from the stool B concentration. So that was another way to look at the how to marry a PBBM and a PKPD, because the PBBM will give you a mechanistic explanation of the dissolution. And for these 3 different batches, actually, there was no PK, but the PD was very nicely matching the anticipated relationship with the drug concentration in the lumen in the cecum. So that was really nice. So in terms of root cause analysis for the delayed disintegration, there was either a salt disproportionation or the issue regarding the disintegrant. And there was lots of methodologies that were conducted to rule out the salt disproportionation. It's really very clear from the analysis that the root cause was a delayed disintegration for the tablet. And that was tested in the PBBM by applying a delay in the disintegration. We use the model to predict the test batch at time 0 and after a certain amount of storage at high temperature. And we compared this test batch results to the pivotal concentration of drug X in the cecum, which will drive the efficacy of the drug, as we've just seen. And basically, we could show that they were going to be comparable. So the conclusions were that this delay in disintegration would not lead to a reduced efficacy of the drug. In terms of take-home messages, I hope I've shown you that PBBM can be very mechanistic and can apply to different physiologies, to different formulations, different dosing regimens. And it's widely used nowadays to bridge formulation. But when you couple it to PKPD, you can actually expand the product quality safe space beyond what is offered by bioequivalence testing. So you can justify more differences, or differences that would not meaningfully impact the safety and efficacy of the drug. And finally, if you have PKPD model coupled to PBBM, and you can use efficacy biomarkers, you can really probe the performance of drugs that was initially really hard to characterize, because for the example of drug X, which is minimally absorbed, you cannot really follow it in the plasma, so it's really hard to validate such a model. But PKPD models can also bring an additional insight into the PBBM performance. And that's the end of my presentation today. I don't know how I'm doing with time, but thanks for your attention, and I'm ready to take any question.
Thanks, Xavier. So we have 2 minutes. We will have time for only one very quick question. And I remember everyone that we are going to have the session, the panel session, where is another opportunity to answer questions with the presenters. So I'm going to take the one from Xiaojun, which is, how do you design virtual batches that are on the border or threshold of bioequivalence? The overall dissolution profile, shapes. So what you can comment on this?
Yeah, it's a very good question. I think you should respect the shape of your reference batch performance. So for example, if you've got a rapid dissolution phase and then a slower dissolution phase, you should try to maybe fit it with a two-phase viable function, and then change the parameters of the viable function so you can reduce the dissolution while still keeping 100% release in the end. So it's and then you can use that virtual B as an input in a mechanistic model, so either by fitting a Z factor or a PPSD on that. But yeah, the shape is important to respect. If you have a non-bioequivalent batch in the clinic and the reference, you can also go halfway. So you can simply average the concentration dissolved at each time point between these batches. So you can create a virtual batch like this as well. That's another option.